get started. Hi everyone, it's really great to see you attending this Head Start lecture. My name is Ivy and I'll be taking you through the content for your final stretch of QCE Biology 3 and 4. I hope that by the end of today you're able to have developed the foundations of the topics that we'll be covering. Strap in because we're in for quite a bit of content, but it will definitely set you ahead in your studies. Now, before we dive into the lesson, let me take a moment to introduce myself and share a bit about my background. Hi everyone, I'm Ivy and I currently work as a tutor at ATAR Notes tutoring service, TootSmart. I have a regular physics class every Tuesday night, so I'm quite familiar with how the sciences are taught in the Queensland curriculum. I actually graduated last year from Clever MacKillop College in Upper Mount Gravatt, where I developed a strong passion for physics and biology, two subjects that really captured my interest. This passion actually led me to pursue a Bachelor of Advanced Science with a major in biomedicine and a minor in physics. My future aspirations lie in the field of molecular imaging, which combines biology and physics together in a really fascinating way. During my time in high school, I also studied methods, literature, study of religion or SOR, and health. While it may seem like a really broad range of subjects, I found that this diversity enriched my understanding and problem-solving skills across different disciplines. I believe that having a well-rounded education not only prepares you for your ATAR, but also lays a solid foundation for your further studies that you might pursue in the field of biology or related sciences. Today's session is not just about ticking boxes for your ATAR. It's about grasping essential knowledge that will benefit you in your future academic pursuits. I remember attending these lectures during my senior year, and they proved invaluable for consolidating my understanding and preparing effectively for exams. This session presents a prime opportunity for you to get ahead in your studies and approach your upcoming assessments with confidence. So, without further ado, let's dive right into today's content. I'm sure you're wondering what we're actually going to be doing today, and that's a really great question. Many of you are probably well into your first topic for Unit 4, which is DNA, genes, and the continuality of life. And because of that, today we're going to be focusing on the second half of that topic, which is inheritance and biotechnology, and actually beginning on our second topic by taking a look at evolution. I'll give you a quick introduction into each of these topics now so that you have some idea of what to expect. Inheritance describes the ways traits are passed down between generations, with genetic information passed from parent to child. Think back all the way to grade 10, where you would have looked at Punnett squares and pedigrees. That is actually the foundation for this subtopic. Next, at its simplest level, biotechnology is exactly what you would expect. Technology based on biology. Basically, we are able to harness cellular and processes to develop technologies and products that help improve our life. Of course, there are a lot of ethical debates around this topic, and we'll also take a quick look at that today. Now, finally, evolution is the change in heritable characteristics of biological populations over successive generations. When I say evolution, many of you probably just think of natural selection, and that is definitely a very important part of evolution. But there's actually a lot more to the story than that, and as you'll find today. We'll be covering a lot of different areas, but they're all really interesting for completely different reasons, so there's a lot to look forward to. Personally, I am pursuing biotechnology, so I have a really keen interest in this topic specifically. So with that, we'll move on. Here's a better breakdown of what we'll actually be looking at today. So, for inheritance, we'll use probability models like frequency histograms and Punnett squares. These tools help us to calculate the likelihood of certain genotypes and phenotypes appearing in offspring based on the inheritance patterns of alleles. Specifically, we're going to be focusing on three types of inheritance: autosomal dominant, where a single copy of a dominant allele leads to the expression of a trait; sex-linked, where genes are located on the sex chromosomes; and multiple alleles, when there are more than two alleles for a particular gene. This last one specifically might be new to you. You might not have learnt about it in junior, so I'll be going in more depth on it. On that note, we're also going to define polygenic inheritance, which involves multiple genes influencing a single trait. Using examples, we'll predict the frequencies of genotypes and phenotypes for traits controlled by three alleles. This will involve understanding how multiple genes contribute to the expression of a trait, such as height or skin color. Obviously, these traits aren't just controlled by 2 alleles. They range on a myriad of spectrums. So we'll be looking at that. By the end of the lesson, you should be comfortable using Punnett squares again and other probability models to predict genetic outcomes for different types of inheritance. You'll also understand how multiple alleles and polygenic traits contribute to the diversity we see in organisms today. Okay, then we're going to move on to our second topic, which is biotechnology. And we're going to be taking a look at the making of recombinant DNA through isolation, cutting, insertion, joining, and amplification. This might sound really foreign to you, but we'll hopefully break it down in steps that will make it clear. We'll then move on to the applications of DNA sequencing and profiling, where we use these recombinant DNA for analysis. Think about true crime shows where they test the DNA to see who the killer is. Basically, we're going to be looking at the biological application of this. This will bring us to PCR and gel electrophoresis, which are 2 really important types of biotechnology. They're really important in the modern day for a bunch of reasons, and we will look at those later as well. And then finally, we'll take a shot at appraising data of genetic biotechnology. This is a really crucial skill to develop if you intend in pursuing higher biology. So it's really important that we understand it right at the moment. That will bring us to our final topic today, which is evolution. In our lesson, we'll cover key aspects of evolution. We'll define evolution, microevolution, which is small-scale changes within populations, and macroevolution, which is large-scale changes leading to new species. We'll also explore episodes of evolutionary radiation and mass extinctions over Earth's 3.5 billion year history. This might also sound a bit foreign to you, but I'm sure later on in the lesson it will make a bit more sense. Additionally, we'll interpret DNA similarity data to reveal phylogenetic relationships among species using comparative genomics to support evolutionary theories. Basically, we're going to look at how species go from one to another. These topics will help us to understand how evolution has shaped life's diversity from genetic changes to major evolutionary events. Okay, so with that, we'll get started by taking a look at inheritance. So, what actually is inheritance? Like I mentioned earlier, inheritance is the biological process through which traits and characteristics are passed down from parents to their offspring. It is governed by the genetic information contained in DNA, which is organized in units called genes. Each gene can have different versions, known as alleles, that dictate specific traits. The combination of these alleles inherited from parents forms an individual's genotype, which in turn influences their phenotype, the observable traits. Understanding inheritance allows us to predict how traits are transmitted through generations, understand genetic disorders, and explore the diversity of life. That's what makes it such a good segue into our other 2 topics, biotechnology and evolution, which tie into this really well. So, before diving deeper into genetics, it's essential to grasp a few key concepts. You've probably heard of these before from year 10, and I mentioned them just before, but they're really important that you have a firm grasp on in order for the rest of this to make sense. So, first, alleles are different versions of the same gene, and they each provide distinct instructions for a specific trait. I like to use a lot of metaphors, so you'll see that, but I like to think of alleles as different flavors of ice cream. They all fall under the category of ice cream, but offer different flavors. Alleles can be dominant or recessive. Dominant alleles exert their effects wherever they are present, while recessive alleles require both copies to be recessive to influence the phenotype. And you would have covered that using Punnett squares in junior years, where you would have been asked to extrapolate the phenotypes of offspring based on the genotypes of parents. And we will be doing that again later today, so hopefully you remember some of it. But for now, I'm going to explore genotype again. Genotype refers to an organism's genetic makeup for a particular characteristic. Essentially, it's the combination of alleles inherited from both the parents. This genetic blueprint determines potential traits, but doesn't always predict how these traits will manifest. For example, the genotype from a dominant allele might be represented as capital A, and for a recessive allele, lowercase a. An individual's genotype could be capital A, capital A, lowercase a, or lowercase a, lowercase a, where capital A denotes the dominant allele and lowercase a, the recessive one. For the sake of this, I'm just going to keep saying big and little as capital and lowercase because it can get a little confusing if I'm saying lowercase, uppercase. Okay, so the phenotype, on the other hand, is the physical expression of the genotype. It's how the trait appears in the organism. So, for instance, the phenotype for blood type A can result from the genotypes AI or AA, where A signifies the specific type. However, phenotypes are not always solely determined on genotypes. Some traits, like skin color, are influenced by both genetics and environmental factors. Sun exposure can significantly alter skin color, and this showcases the interaction between our genes and the environment. There's actually a word for this type of study. It's called epigenetics, and you'll also look at that a bit more later in this topic. So, when discussing alleles, a dominant allele is one that always shows up in the phenotype, even if only one copy is present. It's like having a loud voice in a choir. No matter how many quieter voices or recessive alleles are present, the loud voice or dominant allele will dominate the sound. Dominant alleles are represented by uppercase letters. Conversely, a recessive allele only appears in the phenotype when both alleles are recessive, represented by lowercase letters. In our choir analogy, the recessive alleles are the quieter voices that can only be heard if no dominant alleles are overshadowing them. We also can use the terms heterozygous and homozygous to describe the allelic composition of an individual. So, if an organism has two different alleles for a trait, like big A, little a, it is heterozygous. This is akin to having both chocolate and vanilla ice cream in one bowl. Each flavor is distinct yet present. If the organism has two identical alleles, like two big A's or two little a's, it is homozygous. It's like having a bowl of only chocolate or only vanilla ice cream with no mixture. However, a homozygous dominant individual will look the same as a heterozygous according to Mendelian inheritance. They will both possess the dominant phenotype. And that's really tying in all those ideas together. So to better understand it, however, we're going to explore a specific example involving dogs and a hereditary condition. In dogs, there is hereditary deafness caused by the recessive gene little d. For a dog to be deaf, it must inherit two copies of this recessive gene, one from each parent. So, you can imagine a kennel owner who has a male dog named Gilbert. Gilbert can hear perfectly well, which tells us something important about his genetics. Since the ability to hear is a dominant trait, which is indicated by the gene capital D, Gilbert must have one dominant allele to express this trait. However, we also need to consider two possible genotypes for Gilbert. Since he can hear, he could be homozygous dominant, carrying two copies of the dominant gene. Alternatively, he could be heterozygous, carrying one dominant allele and one recessive allele. But why does this matter? Well, if Gilbert is used for breeding, understanding his genotype helps us to predict the likelihood of his offspring inheriting deafness. If Gilbert is homozygous dominant, all of his puppies will inherit at least one dominant gene from him, ensuring that they can hear. On the other hand, if he is heterozygous, there's a chance that he could pass on the recessive allele to his offspring. If paired with a female carrying the recessive gene, some puppies might inherit two recessive alleles and thus be deaf. So, by knowing Gilbert's genotype, the kennel owner can make informed breeding decisions, minimizing the risk of hereditary deafness in future generations of puppies. Understanding these genetic principles is essential not only for responsible breeding practices, but also grasping the broader implications of heredity and genetics in all living organisms. There is a myriad of applications for this type of knowledge, but is especially useful in things like breeding. So, let's delve into how we can use pedigree charts to predict the inheritance of genetics disorders such as Huntington's disease. Huntington's disease is a type of disease affecting movement, cognition, and mental health. We can represent its inheritance using a pedigree chart like the one shown on screen. A pedigree chart is a diagram that shows the occurrence and appearance of phenotypes of a particular gene or organism and its ancestors from one generation to the next, most commonly used to track the inheritance of genetic traits and diseases in humans. Before we continue, I'm just going to cover some common symbols and conventions. In pedigree charts, squares will generally represent males and circles will represent females. A filled symbol indicates an individual expressing the trait which is affected by the disorder. And a non-filled one will represent one not representing the disorder, and finally, horizontal lines connecting a male and a female represent mating, while vertical lines represent their offspring, lead to their offspring, Okay, so Huntington's disease is an example of an autosomal dominant condition, so just one affected allele is required for it to manifest. When I say autosomal, I'm just referring to genes located on chromosomes except the sex chromosomes. Remember that dominant indicates the presence of one mutated allele overriding the normal allele in determining the phenotype, as seen in Huntington's disease. So, in Huntington's disease, having a mutation in one allele suffices to express the disease phenotype, a dominant trait. The only scenario where an individual does not develop Huntington's disease is if both alleles are normal or recessive condition. Sorry, I just need to clear my throat. I'm losing my voice recently. Okay, I'm just going to go with that last point again because I didn't explain it. The only scenario where an individual does not develop Huntington's disease is if both alleles are normal, which is a recessive condition. So, we can predict the inheritance of these genetic disorders using Punnett squares. These squares depict normal potential genetic outcomes based on parental genotypes. Both unaffected carriers, one affected parent, or both affected parents. For carriers, or heterozygous individuals, the square shows a 50-50 chance of passing on the infected allele. This method is particularly relevant for understanding the genotypic and phenotypic ratios of offspring, clarifying who carries the gene and who displays the disease. We can actually use a similar logic for looking at an autosomal recessive inheritance. Autosomal recessive inheritance is a fundamental concept in genetics, distinct from autosomal dominant inheritance. In autosomal recessive inheritance, the phenotype of interest manifests only when an individual carries 2 copies of the mutated gene, one from each parent. This results in the condition being recessively inherited, meaning it typically appears in homozygous individuals, where both alleles of the gene are mutated. A classic example illustrating autosomal recessive inheritance is albinism. Albinism is characterized by a lack of pigment production in the skin, hair, and eyes, resulting in a pale appearance. For an individual to exhibit the albino phenotype, they must inherit 2 recessive alleles, one from each parent, that disrupt the production of pigment proteins. To understand how autosomal recessive traits are inherited, Punnett squares are commonly used. These squares visually represent the possible genetic combinations that can result from the mating of 2 individuals, showing the possibility of offspring inheriting specific genotypes and phenotypes. In the case of albinism, a Punnett square would illustrate the offspring that need to inherit 2 copies. Illustrate that offspring need to inherit 2 copies of the recessive allele, one from each parent, to express the albino phenotype. The concept of half-half becomes particularly relevant in the context of autosomal recessive disorders. Unlike autosomal dominant disorders, where individuals with just 1 copy of the mutated gene can exhibit the phenotype, recessive disorders often remain hidden as carriers in the heterozygous state. This means carriers do not show symptoms but can pass on the mutated allele to their offspring. Therefore, understanding the pedigree of a family, tracing the inheritance of alleles across generations, is crucial for identifying carriers and predicting the likelihood of offspring inheriting the disorder. You'll more often than not see autosomal recessive inheritance because a disease will generally be the recessive trait. That is because diseases are generally a non-functional protein, so they will get overridden by a functional one. You'll see this a lot more if you explore it at a higher level. But for the moment, grasping these principles through Punnett squares and pedigrees allows you to gain an insight into how autosomal recessive disorders are inherited and why genetic counseling and testing are essential for families with a history of such conditions. This knowledge empowers us to make informed decisions regarding healthcare and family planning, ensuring the well-being of future generations. So, in contrast to autosomal mutations, which can occur on any autosome, X-linked traits are linked to the X chromosome. Females possess 2 X chromosomes, while males have 1 X and 1 Y chromosome. X-linked traits, such as dominant mutations, specifically involve changes on the X chromosome. An example is Rett syndrome, which we briefly discussed earlier in relation to X inactivation previously. Rett syndrome is a genetic brain disorder that typically becomes noticeable between 6 to 18 months of age in females. Symptoms encompass difficulties with language, coordination, and repetitive movements. Often, affected individuals experience slower growth, challenges with walking, and a smaller sized head. Similar to autosomal dominant traits, Rett syndrome manifests when an individual carries just 1 copy of the mutated gene. When predicting the inheritance of X-linked traits like Rett syndrome, Punnett squares are useful tools. This diagram illustrates the potential genetic outcomes based on parental genotypes, considering both unaffected carriers and affected individuals. It's crucial to highlight that Rett syndrome is lethal in males due to their single X chromosome carrying the affected allele, which lacks a corresponding normal X chromosome to counteract the mutation. Is there any evidence in this pedigree chart that Rett syndrome is lethal in males? Have a quick look using your knowledge of pedigrees. Note that in this example, if a female possessed 2 affected X chromosomes, it would also be lethal. It's just because she has 2 that she's able to counteract this mutation. Okay, so X-linked recessive inheritance follows a very similar pattern to what we just discussed. The gene responsible resides on the X chromosome. Females need both X chromosomes to carry the mutation to express the disorder, while males require only their single X chromosome to be affected. Consequently, males have a higher susceptibility to X-linked recessive disorders. An example of an X-linked recessive disorder is hemophilia. Hemophilia is a condition where blood clotting is impaired, leading to an increased risk of severe bleeding from injuries, cuts, or internal trauma, as the blood fails to coagulate properly. This is a very common example because very few diseases actually follow simple Mendelian inheritance patterns such as the ones we're looking at, but hemophilia does because it is caused by protein not being formed correctly. So you'll probably hear this example a lot, and it is a good one to remember because you are expected to remember specific examples of these types of inheritance. So, on the last one I want to look at for our main types of inheritance is Y-linked traits. Now, Y-linked traits are actually exceptionally rare because the Y chromosomes has limited genetic content. If you were to look at a carrier type chart, you would notice that the Y chromosome is much smaller than the X chromosome, so it's not able to contain much information. In fact, it actually only comprises of 63 genes total, which is incredibly small compared to others. Importantly, only males possess the Y chromosome. Therefore, if a father and a mother have a male child, he will inevitably inherit the Y-linked mutation if present. To illustrate the inheritance pattern of Y-linked traits, we can employ Punnett squares. These diagrams demonstrate that all male offspring inherit the Y chromosome, and thus the associated trait from their fathers. This straightforward inheritance pattern underscores the unique transmission of Y-linked traits exclusive through male lineage. Okay, so apart from the typical ways genes can get passed down, there are a couple more cool patterns to explore: incomplete dominance and codominance. These fall under what we call multiple allele inheritance. We're going to start with the first of the two, incomplete dominance. Incomplete dominance is a fascinating genetic phenomenon where neither alleles completely dominate the other. Instead of one allele masking the effects of the other, as in a classic dominant recessive relationships, incomplete dominance results in a blending or an intermediate phenotype of heterozygous individuals. I'll give you a classic example. You can imagine two alleles for a trait, and one might produce red flowers with big R, and the other white flowers with little r. In incomplete dominance, when you cross these pure breed lines, so an individual that is homozygous dominant and homozygous recessive, the offspring, which must be heterozygous, doesn't show pure red or pure white flowers. Instead, they exhibit a completely new phenotype, pink flowers. This occurs because neither the red allele nor the white allele is fully dominant over the other. Rather, they blend together to produce a third unique colour. This blending effect in incomplete dominance is often likened to mixing paint colours. Just as red and white paint can blend to create pink, alleles in incomplete dominance combine to create a new expression of the trait. This blending is not limited to flower colour. It can be observed in various traits among different species. Think when you breed two dogs together, a blonde dog and a black dog, you will occasionally get brown as your offspring, and that is an example of incomplete dominance. But furthermore, incomplete dominance doesn't necessarily mean a strict 1 to 2 to 1 genotypic-phenotypic ratio like classic Mendelian genetics. The phenotypic ratio in the offspring, 1 to 2 to 1, represents a homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Each genotype contributes to the overall spectrum of phenotypes observed, demonstrating the complexity and variability introduced by incomplete dominance. Codominance is another fascinating pattern of inheritance that contrasts with complete dominance and incomplete dominance. In codominance, neither allele is dominant or recessive to another. Instead, both alleles contribute equally and simultaneously to the phenotype of the heterozygous individual. This results in the expression of both alleles directly and visibly in the offspring. A classic example of codominance can be seen in human blood type inheritance. The ABO blood type system involves 3 alleles, I capital A, I capital B, and lowercase I. Alleles capital I and capital A, and capital I, capital B, are codominant to each other, while allele I, lowercase I, is recessive. Individuals can inherit blood types A, B, and O based on this combination of alleles. In the case of blood type AB, both capital B and capital B, A and capital B alleles, are expressed equally, resulting in an individual having both A and B antigens on their red blood cells. This is quite a complex example, but it does show, it's a really common example that you'll see. Another well-known example of codominance occurs in certain breeds of cattle, where coat coloration is determined by alleles that produce distinct colours. For example, in some cattle breeds, the alleles for red colour coat and white colour coat are codominant. Heterozygous individuals display a coat colour pattern where both red and white hairs are visibly present, rather than a blended intermediate colour as seen in incomplete dominance. The concept of codominance extends beyond visible traits like blood type and coat colour. It also applies to molecular traits such as certain enzymes or proteins, where multiple alleles can produce slightly different variations of the protein and that coexist and contribute to the overall function or phenotype. Unlike incomplete dominance, which results in a blending of traits, codominance maintains the distinct expression of each allele. So this characteristic makes codominance particularly valuable in genetics research and breeding programs where precise control and understanding of trait expression is crucial. So it's a common example that you'll see is a cow. In a cow that has incomplete dominance, you would see spotted patterns. You would see a blend of both of the traits, while in codominance you would see each distinct colour. They wouldn't be blending together to form the same colour, they would be blending in spots. Here's a good question to test it, Okay, so in this example, it gives you a good visualization of codominance versus incomplete dominance. You can see here that in incomplete dominance, we have both of the traits blending together to form an intermediate, in this case pink. While codominance, both of the traits are still being expressed, they're just doing so independently from each other. Again, in my cow example, a better idea would be you have a white cow and a black cow. In incomplete dominance, they would blend together to create a grey cow, In codominance, you would get a spotty cow that has patches of black or patches of white, and then the remainder of their coat would be the opposite colour. It's a really interesting phenomenon, and hopefully it can get you thinking about in real life when you might see a flower, what is actually happening on a molecular level. Okay, so we're going to move on to our next one, which is polygenic inheritance. Polygenic inheritance refers to the phenomenon where a single characteristic is influenced by the interaction of multiple genes. So unlike simple Mendelian traits, which is governed by one gene, like eye color in some cases, polygenic traits such as height, skin color, and eye color in humans are controlled by the combined effect of several genes working together. Think about it like this. When you look at your eye, they probably won't look the same as the person next to you, even if we say that you have blue eyes and they have blue eyes, or brown eyes and brown eyes. And that's because it's controlled by multiple traits. But if you look at a cat, for example, their eye colors tend to remain pretty consistent. Again, this isn't a perfect example. They are controlled by multiple, but I'm using it as an example. Another common example you hear about is height. But height is determined by the interaction of numerous genes, which each contribute a small amount to the overall height of an individual. Another example, skin color. Skin color results from the cumulative effects of several genes involved in producing melanin, which is a pigment responsible for skin tone. Finally, yes, again, eye color is a polygenic trait. It is influenced by multiple genes which affect the amount and distribution of the pigment in the iris. That is why you will never see anyone really with the same color as you, because it is so complex. Right now, your understanding just should be that multiple alleles contribute to one phenotype, essentially. The complexity of polygenic traits means that they often display as a continuous range of variation within a population rather than discrete categories. So this variability is evident in human populations, where individuals exhibit a spectrum of heights, skin tones, and eye colors. The contributions of each gene involved in polygenic traits may be additive, where each allele contributes incrementally to the phenotype, or they may interact in a more complex way, such as in epistasis, where the effect of one gene depends on the presence of other genes. But generally, it will be looking at additive examples. So you can essentially think of it, if they have a recessive, they will be on the lowest end of that spectrum. If they have dominant, they will be at the other side. And you can see that really well in this diagram here. But you will know from your studies that if you, even in general inheritance, if you breed a homozygous dominant and a homozygous recessive, or two heterozygotes, you will generally end up with more intermediates than you will exceptions on the end. Think when you breed two homozygous individuals, you get two heterozygotes and two homozygotes, but each are on different ends. One is a homozygous recessive and one is a homozygous dominant. And you can see that happening in this bell curve here. That's a really important idea to understand for this topic. But polygenic inheritance is just a fundamental concept in genetics in general, because it explains how complex traits in organisms are influenced by the interaction of multiple genes. We can apply what we already know onto a wider scale, essentially. Unlike monogenic traits, which are controlled by a single gene and typically follow simple Mendelian inheritance patterns, polygenic traits arise from the cumulative effects of numerous genes scattered along the genome. Going back to our example of human height, while height is influenced by genetic factors, it is not determined by a single gene, but rather a combination of many genetic variants. Research estimates that over 400 genes contribute to variation in human height, and that's why you see such a broad range. Each of these genes plays a small role in determining an individual's stature, with some alleles contributing to taller heights and others to shorter ones. And this results in a continuous spectrum of heights observed in the human population, ranging from very short to very tall, and most individuals falling somewhere in between. You'll notice again that it's following that bell curve that I was describing earlier in this diagram. The complexity of polygenic traits makes it challenging to predict the inheritance patterns in monogenic traits. I don't believe you would be given a Punnett square for a polygenic trait at this level, but you just need to recognize that it follows similar inheritance, just on a more complex scale. So in polygenic inheritance, alleles from multiple genes interact in an additive way, like I mentioned earlier, and this means that the phenotype is not simply determined by the presence or absence of dominant or recessive alleles, but rather the combined effect of all contributing genetic variants. On another note, however, though, environmental factors also play a significant role in shaping polygenic traits, like I mentioned earlier. Factors such as nutrition, health, and lifestyle can influence how genes are expressed, which further complicate the relationship between genotype and phenotype. For example, a child's growth and eventual height can be influenced by their diet during critical growth periods, or their eye color can be influenced by sun exposure. So there's a big relationship between these, and it's endlessly complex. At this level, you're not expected to make the key links in more specific examples, but you are expected to understand that polygenic traits are influenced by multiple alleles, and that these traits can also be influenced by environmental factors, which make them endlessly complex. So to consolidate all that knowledge, I'd like for you to have an attempt at these questions. You're told that to demonstrate polygenic inheritance of colour in wheat, a cross was performed between two intermediate parents. In this case, intermediate is referring to individuals who are essentially in the middle for that trait. They're heterozygous for each. Each dominant allele adds a unit of colour to the phenotype. You're asked which graph would depict the number of individuals and number of phenotypes for the cross. I'll give you a second now to give this a thought. I'm just going to get a drink, and then we'll go through the solution together. Okay, so to solve this problem, we need to understand polygenic inheritance, which, as I mentioned, is where multiple genes influence a single trait. So in this case, wheat colour is controlled by 3 genes, A, B, and C, and each dominant allele contributes 1 unit of colour. So if we cross 2 intermediate parents, which would have the genotype, big A, big B, little b, and big C, little c, we result in offspring that vary with varying combinations of dominant and recessive alleles, ranging from 0 to 6 dominant alleles. So the range of combination typically forms a bell curve or a normal distribution, because there are more ways to achieve an intermediate number of dominant alleles than the extreme numbers. Think back to the example from 2 slides ago, where we looked at skin color. You can see this bell curve and a Punnett square that shows how we get this outcome. Okay. Sorry. So therefore, the correct answer is actually A, because it accurately depicts the expected normal distribution of phenotypes, which would result from polygenic inheritance. You might be fooled to say that B is the correct answer, but as you can see, the 2 intermediate phenotypes are even, so it doesn't accurately display that inheritance that you would expect based on the number of traits. If you got a question like this and you were struggling, you could draw out a Punnett square. Even though it is quite complex, it would give you the general idea. Okay. Sorry, with that last one as well, the reason you might be fooled for C and D, C is showing codominance essentially, and D is showing Mendelian inheritance. But it is talking about polygenic inheritance in the question, so that would be the correct answer. Okay. So we'll have one more question now. You might have sort of sneak at it. This can be a bit of a complex question to answer, so I'll give you a second to have a think about it. You're told that in blood group inheritance in humans, 3 alleles determine blood type, which can be type O, A, B, or AB. It is known that the allele capital I, capital A produces type A and is codominant with the allele capital I, capital B, which produces type B. The allele lowercase I produces type O, and alleles capital I and capital B are dominant over lowercase I. It might be easier for you to read this because explaining it aloud is a little difficult with all the capitals, but you're told that a father who is heterozygous type A and a mother who is heterozygous type B have children, and you need to predict the likely frequency of phenotypes of their offspring showing your working. I'll give you another second to have a think about this, and then I'll go through the solution. Okay, so as we're told in human blood group inheritance, 3 alleles determine blood type, which can be type O, A, B, or AB. Allele capital I, capital A produces type A and is codominant with allele capital I, capital B, which produces type B, where allele lowercase I produces type O. So both our type A and B are dominant over allele I, lowercase I, sorry. So when a father who is heterozygous for type A, a father who is heterozygous for type A will therefore have the genotype capital I, capital A, and lowercase I. The mother who is heterozygous type B is therefore capital I, capital B, and lowercase I as well. Because these are heterozygous individuals, but they're still expressing the trait because it is dominant. If the two have children, the possible combination of their alleles can be predicted using a Punnett square, which I have here. So the possible offspring are type AB, type A, type B, and type O, and these each actually have an equal frequency of 25%, or 1 in 4. So thus, you can answer that question by saying that their children will have a 25% chance of being type AB, a 25% chance of being type A, a 25% chance of being type B, and a 25% chance of being type O. That is a pretty difficult question, so good job if you were able to get that. This is our final question I want to go through for this topic. We'll start with part A. You're asked to describe what is meant by the genotype of an organism. This is just a 1-mark question, so I'm going to just move straight on to the solution. The genotype of an organism refers to its specific genetic makeup or combination of alleles that it possesses for a particular gene. It represents the organism's hereditary information that is encoded by its DNA, influencing its traits and characteristics. Hopefully that wasn't too bad for you, because but if you were able to get it, it's good because it underlies a lot of the concepts we're going through. Okay, I'll give you a second to have a think about the second one, but you're told that inherited mutations can affect the genotype of offspring, and you're asked to describe how this occurs. Think about your response for a second before I go through the solution. Okay, so inherited mutations can affect the genotype of offspring by altering the DNA sequence passed down from their parents to children. These mutations can occur anywhere from germline cells like egg and sperm, and if present can occur later in life, and if present are incorporated into the offspring's genome when they do occur in germline cells. So mutations can be caused by errors during DNA replication or due to environmental factors. If a mutation occurs in a gene, it can change the allele's function, potentially leading to new traits or affecting existing ones. The resulting genotype of the offspring will reflect these inherited genetic changes. Basically what we're saying is that a mutation, if it occurs in the germline, will be present in the offspring, and then this will carry on to successive generations depending on whether or not it is dominant or recessive. That is the general answer. For my live viewers, I will put a better one in the chat because I think that I could explain that better. But for now, that is, if you mention those general things, you'd be good. But we're done with all the information I wanted to go through for inheritance. So good job if you got through that first stretch of information. We're going to go on now to biotechnology, but you can take a quick second now to grab a drink and have a stretch before we move on, because it is a lot of information. Biotechnology is a sort of difficult topic as well, so be prepared. Okay, I'm going to move on for the sake of time, just because I don't want to be under. Hopefully you had some time to get a drink and have a stretch. Like I mentioned, we're going to move on now to biotechnology. So I'll start by asking you an important question. What exactly is biotechnology? Biotechnology is just a cutting-edge field that uses biological systems to create products and processes that benefit society. It combines biology, chemistry, and engineering to manipulate biological molecules, cells, and organisms for practical purposes. A key tool in biotechnology is plasmid technology. We'll learn more about this later, but it's good for you to know right now that plasmids are small circular DNA molecules that are found in bacteria and other cells. They essentially act as carriers to transfer genes between different organisms. This technology allows scientists to insert specific genes into organisms, which modify them, and then they can use that to study gene functions in controlled environments like labs. Understanding plasmid technology is crucial for various biotechnology applications, including genetic engineering, recombinant DNA technology, and gene therapy. These techniques help in producing useful proteins, creating genetically modified organisms with desired traits, and studying genetic diseases. We're going to now explore the basics of biotechnology and focus on their importance in plasmid technology. So at its core, biotechnology is just, like I mentioned, the application of biological systems, organisms, or their components to develop products or processes for specific uses for humans. This broad definition encompasses everything from ancient agricultural practices to the most advanced genetic engineering techniques. To illustrate this, I'm going to consider 3 diverse examples of biotechnology in action that range anywhere from millennia ago to later in the 2000s to now. We'll start by looking at beer brewing. This is actually an age-old practice, and it involves the use of microorganisms, specifically yeast, to ferment sugars and produce alcohol. Here, biotechnology leverages the natural metabolic processes of yeast to create a product that has been enjoyed for centuries. Despite its simplicity, beer brewing is a prime example of how humans have harnessed biological systems to create valuable products. So while the idea of biotechnology feels like it's this whole new thing, we've actually been doing it for ages, as everything is intertwined with biotechnology. We next have penicillin, which is an antibiotic that revolutionized medicine. Penicillin is produced by certain molds, specifically one known as Penicillium notium. And the discovery and mass production of penicillin marked a significant milestone in biotechnology because it utilized the natural properties of mold to combat bacterial infections and has saved countless lives. This example underscores how understanding and utilizing microorganisms can lead to groundbreaking medical treatments. And lastly, a more recent discovery is gene therapy. It is an emerging biotechnology that is aimed at treating genetic disorders. Gene therapy works by delivering functional genes into the cells of a patient to replace or repair defective ones. For instance, in trials for treating cystic fibrosis, researchers have used DNA technology to insert missing chloride channel gene, which is what results in cystic fibrosis, into patients' lung cells. This approach represents the cutting edge of biotechnology, where scientists manipulate genetic material to cure diseases at their source. Of course, this isn't without its ethical concerns, but as I mentioned earlier, we'll be going into those later. These examples demonstrate the diverse applications of biotechnology in everyday life, from the production of beverages and life-saving drugs to cutting-edge medical treatments. But how do these biotechnological processes work at a molecular level? That is where DNA technology comes to play. DNA technology encompasses a variety of techniques that allow scientists to analyze, manipulate, and engineer genetic material. One fundamental technique is DNA cloning, where specific DNA fragments are replicated to produce numerous copies. This process often involves inserting a gene of interest into a plasmid, a circular DNA molecule, which is then replicated within bacteria. We'll be taking a closer look at this in the coming slides as well. But this method is used to produce proteins such as insulin for diabetes patients. Another crucial example that we'll be looking at is PCR, or polymerase chain reaction. PCR amplifies tiny amounts of DNA to generate millions of copies. This technique is essential in various fields, from medical diagnosis to forensic scientists. For example, PCR can help identify disorders or match DNA found at a crime scene to a suspect. And the final one we'll be looking at in the coming slides is gel electrophoresis. which is my personal favourite. It's another important technique that enables researchers to visualize the DNA fragments that they have amplified. By applying an electric field to a gel matrix, DNA is separate based on size, and researchers can stain and view these fragments to analyze genetic material. This method is commonly used to examine the results of PCR reactions. With that, I'll move on to the next. I just want to mention, understanding these DNA technologies is crucial because they form the understandings of many innovations in biotechnology. This drives advancements in medicines, agriculture, environmental science, and it paves the way for solutions to complex biological challenges. So even if you aren't considering studying higher biology, the applications for this type of technology it reaches everywhere, and it's incredibly profound. So I'm going to start by diving into the fascinating world of recombinant DNA technology, which is actually the cornerstone for modern biotechnology. Recombinant DNA is essentially a hybrid DNA molecule, and it's created artificially by combining DNA from one or two or more different sources. This technology allows scientists to produce large quantities of specific genes or proteins, which can then be used in various biotechnological applications, including the development of pharmaceuticals and genetically modified organisms. But to understand how recombinant DNA is made, we're going to go through the steps involved in making it. This can be a bit of a difficult process to remember, so try to focus as I go through it. It's okay if it takes a couple of times to understand fully. But it will also be really helpful for you to have a good understanding of the process of DNA replication from our previous unit if you're struggling to grasp this content. So if you are struggling, I would absolutely recommend reviewing some of the previous stuff. I'm just going to get a drink and then I'll go through the steps. Okay, so let's walk ourselves through this diagram. The first step in creating recombinant DNA is the isolation of the target gene. This is achieved using something known as a restriction enzyme, which is a specialized protein that cut DNA at specific sequences known as recognition sites. They're basically made, you'll learn about restriction enzymes and their purpose in higher level, but just know that they essentially cut DNA. They're able to do that. But they do not cut the DNA through the hydrogen bonds as you would be used to with helicase if you know DNA replication. They cut it through the covalent bonds of the backbone. And these enzymes can cut DNA in two ways. They can do straight down, so resulting in blunt ends, or in a staggered manner, which produces sticky ends. Sticky ends are more desirable for biotechnology applications because they have overhanging sequences that easily pair with complementary sequences on the plasmid, facilitating a more efficient and stable integration of the target gene into the plasmid. When you think about DNA replication, you'll know that having an exposed nucleotide allows another base pair to come and connect to it using hydrogen bonding. And that's essentially what's happening here. It'd be really difficult if you have a blunt end because there's nothing to connect to it, and a covalent bond has to be formed. Okay. So in the next step, we need to prepare the plasmids, which are small circular DNA molecules found in bacteria. Unlike the chromosomal DNA of eukaryotes, plasmids are not essential for survival, but they carry beneficial genes such as those for antibody resistance. So you can think the reason they have these plasmids actually is to transport DNA between other bacterial cells. And this is essentially how bacteria develops immunity to certain diseases that are around it. So these plasmids are used as vectors to transport the target DNA into bacterial cells. The plasmid is cut with the same restriction enzyme that's used to isolate the target gene, which ensures that the end of the plasmid and the target gene are compatible for binding. This creates something that we call a plasmid vector, which is basically a vehicle for introducing new genetic material into a bacterium. The isolated target DNA and the prepared plasmid are often then combined in a solution with DNA ligase, which you'll remember from your previous studies is an enzyme that acts as a molecular glue. It's basically able to facilitate the formation of covalent bonds between the sticky ends of the target DNA and the plasmid. And this results in a recombinant plasmid. This recombinant plasmid now contains the desired gene and is ready for the next step. In the next step, the recombinant plasmid is introduced into bacterial cells through a process called transformation. The bacteria are treated with a solution that makes their cell membranes more permeable, often involving a brief heat shock, and this encourages the bacterium cells to take up foreign DNA, such as our recombinant plasmids that are in the same solution. Now, in order to, not all the plasmid DNA will be taken up, and there's no way that we can actually ensure that it will be. So in order to easily identify which bacteria have successfully taken up the recombinant DNA, the plasmid is engineered to include an antibiotic resistance gene. The bacteria are then grown on agar plates which contain this antibiotic. So only those that have incorporated the plasmid will survive. Basically, it's just ensuring that the plasmid has been taken up. If none of them survive, that means that the plasmid is not there because they are not immune to the antibiotic. In addition, we can also use fluorescent markers which make the bacteria glow under UV light, and that provides visual confirmation of a successful transformation. It's important to note, however, that only 1% of the bacteria will typically take up the plasmid, and not all of these necessarily express the recombinant DNA correctly, and that's why we must use the agar plate. Okay, and the final step can only occur once the bacterial colonies containing the recombinant DNA are identified and have grown. The bacterial colonies are cultured to produce the target protein. So the bacteria transcribe the transferred gene into messenger mRNA or mRNA, which is then translated into a functional protein. After sufficient growth, the bacteria are lythed or split open to release the protein. The protein is then purified through various biochemical techniques such as centrifugation, filtration, and chromatography, ensuring the final product is suitable for intended use. This intricate process of creating recombinant DNA has revolutionized biotechnology because it enables the production of essential proteins and the development of new medical treatments as a result. Now, originally, the reason this was so profound is originally the small size of proteins essentially made it difficult to mass produce them. You could not do it through normal means. But recombinant DNA offered a solution through harnessing the body's pre-existing cellular process. It's a really interesting idea to use what the body has cultivated over millions of years of evolution to our own game. With that, that's a similar diagram showing the same idea. I actually think it might be a better representation of the final step. Sorry, I realized I didn't have that up. But I'll give you a second to take a look at that. Once again, you can see the stages that are happening. We isolate a gene from the human growth that we want. In this case, we've isolated a human growth hormone, and we cut that out using restriction enzymes. We then get a plasmid, and we cut it using the exact same restriction enzymes, and that means the sticky ends are corresponding. That means that when we use ligase, where we add ligase into the same mixture, this gene and the plasmid are able to connect. Then using bacterial transformation, we get this recombinant plasmid to be intaken by the bacterial cell. And then bacterial cells have this really interesting quality where they're able to multiply rapidly. So they do that, and that results in a surplus of the protein which is contained within the bacterial cell. We can then isolate that protein for whatever applications we could use it for. Okay. So we're now going to, this is just another idea. Sorry, I didn't realize there were so many images for this. This is just showing the second process of that part. Once we have isolated the bacteria, this is the process, it multiplying. By including a antibody resistant gene or an antibiotic resistance gene and putting that antibiotic into the plate, we know that the only bacteria that are able to grow are the ones that have the resistance gene because they have uptaken the plasmid. So they multiply and they grow. We get a plate filled with these bacteria that we know have the plasmid that we need. And then we can take off that plasmid from the agar plate and isolate the protein. Okay, so that's all for recombinant DNA, but there's another process we need to break down involving DNA before we move on to our more uses of this recombinant DNA. and that's DNA sequencing and profiling. DNA sequencing is a pretty nifty way to figure out the exact order of A, G, T, and C in a chunk of DNA. So we're really looking at a very low molecular level that even our microscopes wouldn't be able to see. Scientists use these really fancy fluorescent dyes to label each of the building blocks with different colors. They then run this DNA through a gel in a process called gel electrophoresis, which we'll dive into later. And as that DNA moves through the gel, it scans a laser and picks up the colors, which tell us the sequence of those bases. Why does this matter? Well, DNA sequencing isn't just for catching criminals, although it's great for that too. It's used for everyday stuff like identifying people for things like ancestry tests or figuring out the family tree of plants and animals. In forensics, it's crucial for pinpointing who left their DNA at a crime scene, which helps solve cases. Essentially, if you have a strand of DNA and you find the sequence of bases, you're able to identify how closely linked or related two individuals are, because obviously there'll be more commonality between two individuals that have a certain sequence of DNA or based on their parents, as we would have learnt with inheritance. Okay. DNA sequencing also helps scientists to explore biodiversity, like discovering new species or tracking endangered ones. Plus, it's a game changer for understanding how animals reproduce. Like I mentioned, who's the biological parent and who's involved in raising the offspring. There are a lot of interesting applications for this technology, but in order to use it, we do need to understand gel electrophoresis and PCR. So I'm going to look at PCR first because we essentially need to do it in order to do gel electrophoresis. PCR is like a genetic photocopy machine. It's used to make millions of copies of a specific segment of DNA. It is slightly different from recombinant DNA, which we just looked at, and it's the preferable method in the modern age because it is generally faster and does not require as many materials. Again though, it does use an enzyme, and this is DNA polymerase, which acts like a molecular builder joining individual nucleotides together to form DNA strands. This is the exact same polymerase that is responsible for DNA replication. That's why, in essence, PCR is sort of like artificial DNA replication. One of the cool things as well about PCR is the enzyme it uses, which is which is Taq polymerase. This enzyme is really special because it doesn't fall apart at high temperatures because it's taken from hot springs. This quality is really crucial for how PCR plays out. So I'm going to go over the steps involved in it, and you'll begin to see why. The first step is denaturation. In this step, the DNA is heated up, which causes the double-stranded DNA to unzip and separate into two single strands. You can think of it like unzipping a jacket. It breaks the hydrogen bonds holding the strands together. If you take chemistry, you'll know that hydrogen bonds are a lot weaker than covalent bonds. So it makes sense, essentially, that the hydrogen bonds are untied before when you increase it to a certain temperature. And that's why we need to use Taq polymerase, because if we're putting it up to these high temperatures, Taq polymerase would fall apart and denature as well. But it doesn't, thanks to its quality. Next comes annealing. The temperature is lowered to let small pieces of DNA called primers attach to the complementary sequences of the single strand. And as you might know from DNA replication, primers are like the starting points that signal where the copying process should begin. Again, you would have learnt about that in DNA replication. And then the final step is extension. The temperature goes up again to the right temperature for Taq polymerase to get to work. Taq polymerase is, I probably should have mentioned this earlier, Taq polymerase is a type of DNA polymerase. So it starts building new DNA strands from the original ones using the free-floating nucleotides in the mix. These three steps, denaturation, annealing, and extension, just make up one cycle of PCR. So scientists actually repeat this cycle over and over, like hitting the copying button on a printer to make millions of copies of the target DNA segment. It's fast, efficient, and can produce enough DNA to study even tiny amounts found at a crime scene or in ancient fossils. Let me think. Imagine you run this cycle 30 times. If you started with one DNA segment, you would be left with 2 to the power of 30 segments, or over 1 billion. Well, you can see why it might be useful for replicating small amounts of DNA, but why do we care about PCR? Well, it's super handy in forensic for analysing small amounts of DNA from crime scenes. but it's also used in medicine to diagnose diseases by detecting infections or genetic disorders early on. Scientists even use PCR to try and extract and amplify DNA from ancient fossils or extinct species, which could tell us loads about our planet's history and evolution, which ties nicely into our next topic actually. So there's actually a lot of interesting applications for PCR, but oftentimes, like in crime scenes, it's pretty difficult to do anything with a bunch of DNA. You need a tool to analyze it. And that brings me to my personal favorite biotechnology, which is gel electrophoresis. You'll hear people raving about this one because it tends to be a favorite. Gel electrophoresis is a crucial technique for separating DNA fragments based on their size and charge. So basically, gel electrophoresis works like a molecular sieve, where molecules are sorted based on how far they can move through a gel under electric field. This gel is made up from a porous gel material like agarose, or it's just essentially, it can be any material, but generally that is a common one. It needs to be porous, however, in order for this to work. So you can think of this gel matrix as a microscopic maze. Molecules will travel through it, but at different speeds depending on their size. You can imagine that a bigger DNA piece will struggle to get through the same pore as a smaller one, and that's the logic that's being used here. DNA samples are mixed with a loading dye to make them visible, and then these samples are loaded into small wells or slot at one end of the gel. Once the gel is set up, an electric current is applied to this gel matrix. Because DNA is negatively charged, when the current is turned on, it pulls the DNA fragments through the gel to the positive end, which is opposite to where they were loaded. So smaller DNA fragments move faster through the gel because they can navigate through the pores more easily, and larger fragments move slower because they get caught more often. This separation creates distinct bands of DNA fragments along the gel. After they run, the gel is stained with a dye that binds to the DNA. And this makes the bands visible under UV light or other detection methods. Now, you might be thinking, why is this so useful? And why is it such a favorite? Well, for one, gel electrophoresis is crucial in forensic scientists for DNA profiling. By comparing the band patterns of DNA samples, like those from crime scenes and suspects, scientists can determine matches or exclusions, helping solve crimes and identify criminals. For example, this example on the slide, you can see that this is the DNA found at a crime scene, and these are the DNA from the suspects. The band for the suspect 2 aligns with the crime scene, which suggests that that's their DNA, and suggests that they're guilty. You'll probably do a couple of examples in class for this, but it's a fun and interesting one, especially with, I know a lot of people that have a keen interest in forensics. It's actually also used in genetics to study polygenic inheritance, where traits are influenced by multiple genes. By separating and analysing DNA fragments associated with specific genes or regions, researchers can understand how traits are inherited across generations. The final one I want to mention, in food science, gel electrophoresis is used to test the purity and quality of products like champagne. It can also detect genetically modified organisms by analysing the DNA present in food samples, which ensures food safety and regulatory compliance. As you can see, there is a whole wide range of applications for this biotechnology, and extends into other things besides just biology, which is what makes it such an interesting one. Okay, let's try a question to consolidate this knowledge. You're first asked to state 3 components of a DNA nucleotide. This is more recall from your previous studies rather than what we just talked about, but it's important about what we covered today in order for you to gain something. I'll give you a second to consider your answer. Okay, so a DNA molecule is composed of 3 key components: phosphate groups, deoxyribose sugars, and nitrogenous bases, because this is a 1-mark question, you would receive the mark just for stating that, but I'm going to go a little more in depth for the sake of the ideas we've been covering today. The phosphate groups and deoxyribose sugars form the backbone of the DNA structure, linking together to create a stable chain through phosphodiester bonds. Attached to each deoxyribose sugar is a nitrogenous base, which carries genetic information. There are 4 types of nitrogenous bases in DNA: adenine, thymine, cytosine, and guanine. These bases pair specifically A with T and C with G through hydrogen bonds, enabling the double helix structure of DNA. And this idea is really foundational to all of the biotechnologies we've been discussing. The second question is asking you to describe the steps involved in DNA profiling. So I want you to consider the parts of a DNA molecule in response, DNA nucleotide in your response. That's why these two are a part of the same question, even if they sound like they might be a bit unrelated. I'll give you a second to do this. Okay, so DNA profiling involves several steps to identify and compare genetic material from different individuals. First, DNA is extracted from cells and biological samples such as blood, saliva, hair, or skin cells. Next, we use something called short-chain repeats, which are highly variable regions in the DNA. You aren't expected to explicitly refer to that if you just have an idea of the fact that we isolate a specific region of DNA. That's good. And these are targeted and amplified using PCR. The structure of a DNA molecule underlies PCR, so if you'd like to go more in depth, that would be a great idea for this question. So you could discuss how the base pairs are separated and then amplified using primers and Taq polymerase. And then this process produces millions of copies of the specific DNA region, which allows for DNA detailed analysis even from small DNA samples. The amplified regions are then separated using gel electrophoresis, which applies electric field to a gel matrix, resulting in distinct patterns of bands for each individual. And then finally, the DNA band patterns are compared between samples to determine genetic similarities and differences. This actually allows for precise identification and comparison of genetic material, which is what makes DNA profiling such a powerful tool in forensic scientists, paternity testing, genetic research, and a myriad of fields. This is a slight change in direction, but I do want to address this before we move on. You'll see this cognitive verb a lot, appraise, when you're discussing biotechnology. When we talk about appraising a technique, we're talking about looking at how it works. This is really crucial if you decide to discuss biotechnology in any of your research investigations. So you can imagine appraisal as you've got this new, I'll use an example actually. Imagine you've got this new way of doing things, whether it's a method for growing plants faster or a treatment for a disease. You want to figure out if it's actually doing what you hoped it would. So first off, you would set a couple of groups to test it out. One group gets the new technique, and that's your treatment group, and the other doesn't get anything different, and that's your control group. Then you could watch closely and measure things like growth rates or how much of a task they've completed. And you can think of it like checking how far along they've come. Once you've got those numbers, you can compare between the treatment group and the control group. And this comparison tells you if the new technique is really making a difference or if it's doing the same thing as the previous. Based on what you find, you can make smart decisions about what comes next. Maybe the technique really boosts growth rates, so you say, hey, this could be the future of farming. Or maybe it doesn't do much from what's already out there, and you might need to tweak it a bit before it's ready for use. So, really, appraising is about testing something out, seeing how well it stacks up against what's already known, and then deciding whether it's worth pursuing further. In our case, we could also use it for appraising the outcomes from gel electrophoresis. We're comparing what we already know against what we have using a technique, and then from that evaluating a conclusion. For example, going back a couple of slides, we could say we had a crime scene and the question was asking about that. Our appraisal would be using amplifying the DNA using PCR, and then using gel electrophoresis to get bars that show us the genetic info for an individual. So that's a really crucial skill that will underlie biotechnology, especially if you decide to pursue it further. And another quick change of pace, but a very important thing to mention that I've been going on and on about is bioethics. Bioethics is a big deal in fields like medicine and health sciences, especially when it comes to biotechnology. Biotechnology has the potential to do a lot of good, and don't get me wrong about that, like improving health and solving problems, but it also comes with a lot of risks and tough ethical questions, just like any new tech. So one significant example where bioethics was violated in the past was the Tuskegee syphilis study, which was conducted in the United States from 1932 to 1972. You're not expected to know this example. I just think it's a really good idea of how violations can hurt not only the credibility of science, but also the long-term repercussions they can have on the community. So in this specific study I'm talking about, the US Public Health Service recruited 600 African American men, approximately 400 of whom had syphilis, while 200 did not, and these were the control group. The men were told that they were receiving free healthcare from the government, but in reality they were not informed of their diagnosis and were not provided with adequate treatment for syphilis, even when penicillin had become widely available as a cure for it in the 1940s. This study was conducted to observe the natural progression of untreated syphilis in African American men, and it came with their uninformed consent and without regard for their health or well-being. The study actually continued for 40 years despite mounting evidence of the harmful effects of untreated syphilis and the availability of a cure at the time, and it wasn't until 1972, which is actually pretty recent, all things considered, that a whistleblower exposed the unethical nature of the study to the public. And that's when it was halted. It had continued up until that point. The Tuskegee syphilis study became a notorious example of exploitation and injustice in medical research, and it led to significant changes in research ethics regulations and guidelines to protect humans. This isn't necessarily related to biotechnology, but it is related to the field of bioethics. It's about whether we have an ethical morality as scientists and people studying scientists to do things for the sake of research or do things for the sake of public health, especially in biomedicine. But looking at it through a biotech lens more now, biotech needs to go through rigorous testing and government checks to make sure it's safe and that it works well because of all the concerns that arise from it. Sometimes though, even with that, problems can pop up and things might need to be taken off the shelves, like certain meds. Ethical concerns in biotech go beyond safety. You can imagine that finding out that your health insurance might charge you more because of a gene that you inherited could be quite an issue, or that you could have access to, if you got your genetic information sequenced, like your entire genome, having that information could be potentially dangerous depending on whose hands it is. Would you be comfortable with your school or your boss knowing about your entire gene, the entire being that makes up you? There are a lot of real life dilemmas that biotech brings to the table. There are also a lot of environmental concerns, like genetically modified organisms in crops help cut down on pesticides, but they also mess with local plants or wildlife if their genes spread where they shouldn't. As you can see, this bioethics ranges into a lot of different categories, but at the heart of it is really just the idea of, is it really ethical for us to use biology, and then is it ethical for us to use that information that we can obtain from biology in a public health setting? If you do continue to study biology, you'll hear a lot about this topic. Okay, but with that, we're going to move on to our final topic, which is evolution. Like I mentioned earlier, you might be inclined to assume evolution is just natural selection, but there's actually a lot more to it than that. You can imagine a population of animals living in a certain environment. Over generations, random changes or mutations occur in their DNA, and some of these changes might give certain individuals a slight advantage in surviving and reproducing in their environment. These advantageous traits get passed down to their offspring, while less advantageous traits may become less common or disappear. As this process occurs over thousands or even millions of years, These accumulated changes can lead to the formation of new species that are distinct from their ancestors. And this is the idea really of natural selection in terms of evolution. It's probably what you're more familiar with. But evolution is really just the story of how life on Earth is transformed and diversified from simple organisms to the vast array of plants, animals, and microorganisms we see today, and then how this continues to happen today on a smaller scale over shorter time. In this example, like the previous example I gave, we're picturing evolution of life over a geological time span. So it's about the major transformations that occur over millions of years, resulting in the emergence of entirely new groups of organisms. For example, from a single-celled organism to complex multicellular life, or from reptiles to mammals. This is actually an example of something called macroevolution. Macroevolutionary changes are often associated with significant events in Earth's history, such as mass extinctions or the colonization of new habitats. New groups and species emerge, adapting to changing environments and ecological niches. However, we can also zoom in on specific species or populations and look at evolution from that perspective, and this is known as microevolution. Microevolution focuses on the smaller scale changes of the genetic makeup of individuals within that group. It focuses on processes like natural selection, genetic drift, or gene flow that can alter the frequency of alleles, or different versions of genes, over relatively short periods of time. Compare this to macroevolution, which looks at things over very large periods of time. This is much smaller scale. These changes don't necessarily lead to the formation of new species, but rather affect the genetic diversities and characteristics within a specific population. It's like observing how different traits become more or less common in a group of animals or plants over a few generations by chance or immigration. But together, evolution, macroevolution, and microevolution provide a comprehensive framework for understanding how life has evolved and adapted to ever-changing conditions on Earth. And that's what we'll really be looking at for the remainder of this lesson. If you're struggling to grasp the idea of micro versus macroevolution, this diagram shows it pretty well. Here on the left you can see macroevolution. You can see that over time taxonomic groups diverge and new species are formed. This takes a very long time to occur over successive generations, and you would not observe these changes over your lifetime unless it's you generally wouldn't. Now, on the other hand, microevolution just refers to small changes to allele frequencies within a species. This is often due to random chance or migration or immigration between two populations of the same species. Now, it's actually up to your personal debate whether you agree with this diagram or not in terms of the microevolution it's given, because a lot of scientists or students would argue, including me, that microevolution really only occurs within the same taxonomic group. That's essentially the definition. So you can imagine that this very fox-looking animal here is just a dog, and these are all Canis lupus. Otherwise it might get a bit confusing if you're trying to think, at what point does a fox differ enough from a dog to become microevolution? But yeah, it does get the general idea across that you can observe several different types of alleles in a population of the same species, and that's microevolution. So, as we've found, evolution is the gradual change of genetic makeup, physical traits, and behavior of organisms over generations. These changes can be significant enough that descendants may become so different from their ancestors that they can no longer interbreed, leading to the emergence of new species. That is generally what we define a new species as, if you recall, as an animal that can no longer interbreed with another member of its original same species. But to understand evolution, we need to consider the vast timescales over which it occurs, especially on a macroscopic scale. So Earth is about 4.5, 3.5 billion years old, depending on your source, and for its early history, around the first billion years, life likely did not exist. Scientists have developed the geologic timescale to organize Earth's history based on rock layers and fossils found in them, and this scale is divided into eons, which are roughly 1 billion years long. The main eons are the Archean, Proterozoic, as I believe it's called, and the Phanerozoic. You wouldn't be expected to remember those names, so don't worry too much about it. It's just getting across the general idea that Earth's history can be separated into eons. Okay, I'm going to focus on the last one, the Phanerozoic. Sorry, I'm going to butcher that name. eon, and that's the most recent one. But we can divide that more into eras as well. The Paleozoic, the Mesozoic, which is when the dinosaurs lived, and the Cenozoic. Again, you're not expected to memorize these, but I just want to re-emphasize the idea that we can break up Earth's history into certain times based on time and use that as a scale for evolution. The Mesozoic and Cenozoic eras are then divided into periods such as the Triassic, Jurassic, and Cenozoic. The Xenozoic era is divided into tertiary and quaternary periods, including the Paleogenic and the Neogenic. And the quaternary period includes the, I'm going to keep botching these names, the Pliocene and Holocene epochs. So while it's not necessary for you to obviously memorize all these periods, understanding them helps in interpreting periods of evolutionary changes and mass extinctions. And also gives you an idea of how little of time we've really had on this planet compared to its actual lifespan. So I'm just going to get a drink. Okay. So, oh my dear, I'm losing my voice over this lesson. Okay. So evolutionary radiation is another concept that you're expected to understand. And it refers to when, I'm just checking that we'll cover this later. Okay, refers to when, I will be introducing this more later. But it just refers to when new species rapidly diversify, which fill ecological niches left vacant by mass extinctions. We can often, this is due to the evolution of new traits or adaptations that lead to them getting advantages in their environments. We often use phylogenetic trees or cladograms, which are diagrams that are used to compare species evolutionarily. And they show how species are related through their common ancestors, which helps us to understand their evolutionary histories. So scientists use various methods to compare DNA between species, such as DNA sequencing and DNA hybridization, as we learnt earlier. And DNA sequencing, which we covered, reveals genetic similarities by analyzing DNA sequences directly. And that helps us to understand how closely related these types of organisms are. Microevolution involves small-scale changes in allele frequencies. You can observe that here. This actually only shows macroevolution, which is the formation of different taxonomic groups over time. It's not a completely accurate example. This would be more resemblant of a cladogram, because a cladogram doesn't show geological time, but we'll look at that later. I just want to give you an idea of how our evolution has changed over time. So as you hopefully know by now, evolution is the process by which populations of organisms change over time, and is driven by several key mechanisms that change and shape genetic diversity and adaptation. And there are actually 4 main mechanisms that you're expected to memorize. These are mutation, gene flow, gene drift, and natural selection, which each play a crucial role in the dynamics of species. Okay, so we'll start with mutation. Mutation is where genetic variation originates. It happens when there are changes in the DNA sequence of an organism. Mutations can arise spontaneously during DNA replication or due to external factors like radiation or chemicals. Most mutations are neutral or harmful, but occasionally they provide beneficial adaptation that improves the organism's fitness in its environment. For instance, a mutation that grants resistance to a disease or allows an organism to exploit a new food source can boost its chances of survival and reproduction. On the other hand, gene flow refers to the movement of genes between populations of the same species. This occurs when individuals migrate or interbreed with members of another population, transferring the genetic material. Gene flow reduces genetic differences between populations and introduces new genetic variations. It helps maintain genetic diversity between populations and promotes similarity within populations, and promotes similarities between populations. On the other hand, gene drift, which is very easy to get confused with, but there are different ideas, describes the change in allele frequencies due to random events rather than adaptive pressures. So unlike natural selection, genetic drift is a random process that can reduce genetic diversity, particularly in small populations. Two main forms are the bottleneck effect, where a population dramatically shrinks due to events like natural disasters, and the founder effect, where a small population starts a new population with limited genetic variation. A key difference, so essentially gene drift is random. Essentially say that there's an ant population, someone came over and stepped on them. They were all contested in the same area and it was independent, they did not intentionally try to step on them. The ones that are stepped on are random, but by chance you could have a selection of alleles on the left side and a selection of alleles on the right that are different. The alleles that are on the left have survived and are therefore more likely to be in higher quantities. But this wasn't due to natural selection, it was just by chance. And that brings me to natural selection. This is probably the one you might be the most familiar with. Natural selection is the mechanism that drives adaptation through differential survival and reproduction of individuals with advantageous traits. It operates in various modes, like stabilizing selection, which favors intermediate phenotypes, directional selection, which shifts towards one extreme, and disruptive selection, which supports both extremes over intermediates depending on environmental pressures. And you will investigate these a lot later in the year as well. But the main takeaway to have about natural selection is that essentially it is intentional selection against a certain trait. Say you have a mice population and the sand is ashy grey. You have a grey mouse and a black mouse. The grey mouse is more likely to survive because prey are more likely to notice the black mouse. In this example here, the frog, it's not the best example, but you can see that the frog clearly notices the ones that have spotted patterns on them more obviously than the regular plain red ones. So it naturally eats those ones, and that results in more plain red individuals in the population. These mechanisms, mutation, gene flow, genetic drift, and natural selection, all interact to shape how organisms evolve over generations. They explain the incredible diversity on Earth and how species adapt to their environments through continual evolutionary processes. They can affect anywhere from a microscopic to a macroscopic level in terms of evolution, and that's what makes them so interesting. This brings me to the next concept I did briefly introduce earlier, which is evolutionary radiation, which is also known as adaptive radiation. And this is a phenomenon where a single ancestral species rapidly diversifies into a multitude of new species, which are each adapted to different ecological niches. This process is driven by various factors such as climate change, geological shifts, atmospheric variations, and ecological opportunities. For example, after the extinction of dinosaurs, mammals underwent an adaptive radiation or evolutionary radiation, in which they diversified into various forms to occupy different ecological niches which were previously dominated by dinosaurs. As you might be aware, I'm going to shift again, but as you might be aware, extinction refers to the complete disappearance of species, where there are no longer any individuals left alive. It's a natural process that's been occurring throughout Earth's history, often due to environmental changes or competition with other species. But human activities have significantly accelerated acceleration rates in recent times, primarily through habitat destruction, pollution, and climate change. That is the general idea, and that's how it links to the idea of evolutionary radiation, because generally evolutionary radiation will follow an extinction event because of all the new niches that are available to occupy. On the other hand, taking that up a notch, mass extinction events are catastrophic events in Earth's history where a large number of species go extinct over a relatively short period. These events are evident in the fossil record and are typically caused by major environmental disruptions such as asteroid impacts, as is the dinosaurs believed to be, extreme volcanic eruptions, or rapid climate changes. The most famous example is the Cretaceous-Paleogene event, I believe it's called, as I have here, extinction event, and that's actually just the one that wiped out the dinosaurs, which and many other species around 66 million years ago. And the most likely theory as to what it was is an asteroid impact, but of course there's a lot of ideas. Okay, so we actually can say in a way that evolutionary radiation is, I wouldn't, the opposite of mass extinction is a bit of a bad example, but they're sort of intertwined but opposites. Like we learned, evolutionary radiation is characterized by the rapid diversification of species into a variety of forms and the abundance of new ecological opportunities such as changes in climate, geological formations, or the colonization of new habitats which provide the conditions for rapid speciation. In contrast, a mass extinction event is marked by a sharp decline in biodiversity, where a significant portion of Earth's species are lost over a relatively short geological time period. These events often result from catastrophic environmental disruptions like asteroid impacts, volcanic eruptions, or dramatic shifts in climate. And these mass extinctions often lead to the abrupt disappearance of numerous species, which cause ecological upheaval and the restructuring of ecosystems. In essence, evolutionary radiation is the rapid increase in diversification, while mass extinction is the rapid decrease in diversification. But there's essentially, you can think about it like there's a threshold for diversity. So once all the niches are occupied, if you've learnt about niches yet, they're sort of just the roles within an ecosystem. So once all the roles are occupied, new species are less likely to be formed because they can't really inhabit a new role. But when a mass extinction event happens, all this diversity decreases and allows an opportunity for this to happen. Okay, and this is our final topic, I believe, for today. In order for you to understand phylogenetic relationships, I need to start by defining molecular homology. Molecular homology refers to the similarity found in DNA and protein sequences among different organisms, and it's a really fundamental concept in evolutionary biology because similarities in these molecular structures suggest a common ancestry. When species share highly similar DNA or protein sequences, it indicates that they likely diverged from a recent common ancestor. In evolutionary terms, recent could span hundreds of thousands of millions of years ago, depending on the species and the rate of genetic change over time. Comparative genomics, on the other hand, involves the comprehensive comparison of genomic features across different organisms. So this includes examining entire genomes, individual gene sequences, regulatory sequences, or the presence of gene duplications and the occurrence of non-coding DNA and repetitive gene sequences. By analysing these genomic characteristics, scientists can discern evolutionary relationships and infer evolutionary histories among species. The more genomic features two species share, the more recent their common ancestor is likely to be. Phylogenetic classification utilizes the information gathered from molecular homology, which you'll remember is the similarity found in DNA between different organisms, and comparative genomics, which is the comprehensive comparison of them, to classify organisms based on their presumed evolutionary relationships. Really, phylogenetic classification is sort of the umbrella term for both of these combined together. And we can do this using a phylogenetic tree or a phylogram, which visually represents these relationships. It's a diagram that illustrates how species are related and how they've diverged over evolutionary time. In a phylogenetic tree, the length of horizontal lines represents connecting species reflects the genetic distance between them. Shorter distances indicate closer genetic relationships, while longer distances signify more distant relationships. One sec, I'm just going to get a drink. Okay, and when the horizontal lines in a phylogenetic tree are not scaled according to genetic distance, it's referred to as an unscaled tree or a cladogram. Cladograms focus on showing the branch patterns and relationships among species without emphasizing the exact amount of time between the genetic change. They're particularly useful for illustrating evolutionary relationship based on shared derived traits rather than genetic distance alone. So if we look here at this example, you can see here that the example on the left would be a phylogenetic tree, while the example on the right would be a cladogram. Now, generally you'll have a key on the bottom which shows the difference. Sometimes this can be in terms of time. It'll, in this case actually they've used alleles or base pairs as a way of showing the difference, but essentially it's the same idea either way. You can see, I'll give an example. Using our phylogenetic tree, if we look at the Pecaria compared to the pig, there is a base pair difference of only 2, because you can look at the difference between the split and identify that it is 4 minus 2, which is 2. And then it's comparing, you can add these up essentially in order to determine the base pair difference from the original organism. So the common ancestor that the pig, Pecaria, and camel share, we could say that there is a 7 base pair difference between it, the Pecaria and the ancestor. There is a 5 base pair difference between the pig and the common ancestor, and 8 base pair difference between the camel and the common ancestor. On the other hand, phylogenetic trees, the cladogram shows a very similar idea. This is exactly, it's a unrooted tree, sorry, I didn't realize. I was just reading based on the cards. But this is actually referred to as an unrooted tree. So it doesn't show it as a horizontal thing. It's a very similar representation. The common ancestor is just represented at this intercept part in the middle instead. But a cladogram is just very similar to a phylogenetic tree. It just doesn't use a key generally, and it just shows the relationships instead. Okay. To consolidate, I want to give you guys a quick break before we move on. I know there's been a lot of information and we are near the end, so just take a minute now if you'd like to have a quick stretch, and then we'll move on. Okay, so we're going to move on to doing, I think there is 2 questions we'll have a shot at. To consolidate all that info about evolution, we're going to go through a few final questions. First, you're asked to explain microevolutionary change. This is a 4-mark question, so you should take that in consideration with your response. I'll give you a minute or 2 to have a think about it, since we do have the time for you to have some time to actually think about these answers, and then I'll work through the answer with you. Okay, I will go through the solution now. Hopefully you had some time to think about your answer, even if you didn't write it out or anything. But I'll just go through what I would personally say for this. Microevolutionary change through mutation involves small-scale alterations in the genetic makeup of a population, which is driven by random changes in DNA sequences. Mutations are really the ultimate source of genetic variation, introducing new alleles into a population's gene pool rather than just pre-existing ones. So here's how the process works. Mutations can occur in various ways, such as errors during DNA replication, exposure to certain chemicals, or radiation. These changes can affect single nucleotides, known as point mutations, large segments of DNAs, such as insertions or deletions, or even entire chromosomes. Many mutations are neutral, and they have no immediate effect on an organism's fitness. However, they can also be harmful or beneficial. Harmful mutations can reduce an organism's chance of survival and reproduction, which often leads to a decrease in frequency over time. This is by the process of natural selection, which is a part of evolution, as we would have learned. Now, on the other hand, beneficial mutations can provide an advantage, such as an increased resistance to disease or better adaptation to environmental conditions. When a beneficial mutation arises, individuals carrying this mutation may have a higher chance of surviving and reproducing as a result. This means that they pass this advantageous allele to their offspring, which gradually increases its frequency in the population over successive generations. This process can lead to significant changes in the population's genetic structure. Even though mutation occurs randomly, the impact on the population is shaped by natural selection. Beneficial mutations are more likely to persist and spread, while harmful ones are typically weeded out. Through this mechanism, mutations contribute to their microevolutionary changes that drive the adaptation and evolution of species over time, and hence macroevolution. Hopefully you're beginning to see why this is just an extension of this question, but hopefully you're beginning to see why we can't really classify evolution as just natural selection. There's a lot of intertwining elements that go into evolution, that combine all these elements such as mutation, natural selection, gene flow, gene drift. So hopefully this question is really just consolidating that understanding. This is the next and final question I wanted to work through with you. It's a little bit shorter, and you're asked to explain a possible consequence of population bottlenecks. This is a really common one to get on externals and things. They love to throw in a bottleneck question. So have a shot and then we'll work through it together. We do have some time at the end of the lesson, so I'm going to try go through some of the ideas that I think can be a bit more difficult to grasp the first time around, since as always you've probably learnt that biology is a subject that's best learnt through repetition. Otherwise I can quickly go through as well some of my personal thoughts about how to tackle this topic, and then we'll hopefully be done. Otherwise, yes, I'll give you a quick chance to have a shot at this question before we do that. Okay. So, it's always good to start answering explain questions by defining the key term in the question. Explain, when I say that, I'm referring to the cognitive verb that the question is asking. Sure, a lot of schools will pump into you understanding the cognitive verbs, and it is very good practice to get into. So, a population bottleneck occurs when a population size is dramatically reduced for at least one generation, often due to an environmental disaster, disease, or human activity. And one significant consequence of a population bottleneck is a loss of genetic diversity. Of course, I'm explaining what the actual concept is now in terms of that cognitive verb. So when only a small number of individuals survive to reproduce, the gene pool of the post-bottleneck population contains much less variation. This reduction in genetic variation can hinder the population's ability to adapt to new environmental changes or challenges such as emerging diseases or climate changes, potentially compromising long-term survival. Additionally, a smaller population size often leads to inbreeding, where closely related individuals mate with each other, increasing the likelihood of inheriting harmful recessive traits and resulting in a higher incidence of genetic disorders and reduced overall fitness. The effects of genetic drift are also more pronounced in small populations, which leads to random changes in allele frequencies that can further reduce genetic diversity and result in the loss of beneficial traits. Now, you wouldn't be expected to do this, but I'm going to use an illustrative example to further my point. You might have heard about this because I believe there was a documentary about it by David Attenborough, but the northern elephant seal population was reduced to, I believe, as few as 20 individuals in the 19th century due to hunting. Despite a recovery in population size, the genetic diversity of northern elephant seals remains much lower than that of southern elephant seals, which makes them far more vulnerable to diseases and environmental changes. So what do you think about it? This is a good idea to start considering as well, because you may be asked about this. There's potential strategies that can be implemented to prevent this reduction in genetic diversity, such as outbreeding. And by that I mean breeding these northern elephant seals with the southern elephant seal population in order to increase the genetic diversity. Because if you were to continue breeding this population, it would result in extremely small population of genetic diversity because and increased frequency of mutations due to inbreeding. But really, in summary, a population bottleneck can lead to significant genetic variation loss, which increases inbreeding risks, reduces adaptability, and makes the population more susceptible to extinction. Okay, so before I think I'll go back before I give my closing statements. Let me go through these topics. These will come naturally to you through practice. The idea of which are recessive and which are dominant. I'm sure you actually have a pretty familiar idea thanks to junior biology. So that should hopefully not be too bad. Incomplete codominance is and incomplete dominance. Incomplete dominance, it sort of sounds like in the name how you would expect it to. Codominance suggests they're cooperating. You can think of other words that start with co. They are together. They're not trying to fight each other essentially. So that means that they both appear together. They're allowed to exist. While incomplete dominance sort of suggests that they're incomplete. They're not overriding each other. They are overriding each other technically. They're sort of fighting to see who appears. And by doing that, they mix together. Again, this is another thing that will probably come to you better through frequent revision, but at the moment having that idea is really good. Okay, I'll move on. Again, you're not expected to, I believe, know how to do a polygenic inheritance chart. They're more expecting you to understand the basics of this idea. And a good question, yes, would be you'd be given that graph and then asked to identify what type of inheritance is happening, and the answer would be polygenic because you can't receive more than 2 different types of phenotypes in typical Mendelian inheritance. Okay. Again, the blood type example, it's an extremely difficult example to deal with because it requires you to actually look back and think of infectious diseases, which would have been your last topic for Unit 2, and your knowledge of antigens and antibodies. I would say there's a good chance many of you have already forgotten that. So I generally, even though it is assumed knowledge, they wouldn't expect you to apply it. But again, you just need to be able to recognize these types of things anyway. Just follow what you know from this unit and you should be able to answer these types of questions, alright. Again, biotechnology is a relatively difficult one, but it will come to you through practice. I'll take a look back at this real quick because now that you have a bit of a knowledge of what gel electrophoresis is, it should make more sense. Essentially, when these, we complete PCR, in which allows us to get a segment of DNA amplified. and then we put it through gel electrophoresis to get bars. And essentially we're doing it, it's a relatively difficult concept, and I actually don't see it turn up on the exam very often, DNA profiling. But it's the idea that you should have really is that they're read, you're able to read base pairs using gel electrophoresis in order to get a sequence of the DNA on a molecular level. They're more expecting you to know that because a lot of the time you will be doing practicals for gel electrophoresis and PCR depending on the materials your school has. In fact, some students go to UQ, I believe, to do it there. But the general idea you should have is just, yeah, you can read the base pairs using biotechnology. Okay. So that about sums up all the main information. Again, this will come to you a lot through consolidation, which is why it's good that you've attended today, because it's really set yourself up well to start ahead, since biology in general is a subject that relies heavily on you being able to actively recall. With that, I'll give my closing statements a bit. So a huge thanks for attending today. I hope that the information was helpful for you and in finding success on the rest of your senior biology journey. There's going to be plenty more lectures these holidays run by ATAR Notes, so if you haven't already and you found today useful, or you think that you might benefit from doing some other subjects, I would absolutely recommend checking them out. This is just the subject I'm teaching, biology. My co-workers are also teaching the other ones, so I'd absolutely recommend taking a look at them. On the other note, I just want to make sure you have some time for yourself these holidays. It's really great that you're attending these lectures, and you're definitely setting yourself ahead, but make sure that you give yourself a break, because senior can be stressful and you won't perform your best if you're constantly overwhelmed with work. I know personally senior can get very overwhelming, especially since you're going into the last term now. From experience, I think that the biology from here on out tends to get a bit easier. So you're going to be looking now at more what we're looking at later in this lesson, which is evolution and ecological biology rather than molecular, such as DNA replication. And from my personal experience, a lot of people tend to agree that it gets easier. I don't want to make you get the idea in your head completely, but if you're feeling overwhelmed, it's okay, don't worry, 100%, it happens to everyone. With that, I really hope you have an amazing rest of your holidays. I really appreciate you coming today. I'm going to end up the recording now. So thank you so much. I hope you have an amazing holidays again. And thank you so much for listening. I hope you have a good day.